You have a vote.
Okay. So thank you all for coming today. Today I'm just going to be talking a little bit about my project regarding the effects of estrogen on von Willebrand factor regulation. Or not. Oh, here, okay. So von Willebrand factor is an adhesive plasma glycoprotein that's transcribed in both endothelial cells, which are the cells that form the inner lining of the blood vessel walls, as well as megakaryocytes, which are platelet precursors. However, the majority of VWF is made by endothelial cells. And in these cells, it gets translated into a pre-pro polypeptide composed of a signal peptide, a propeptide, and the mature subunit. And so the signal peptide helps direct the structure to the endoplasmic reticulum, where it can begin to dimerize. And subsequently in the Golgi, it can multimerize into structures ranging from 500 kilodaltons, which would be low molecular weight VWF, all the way up to 20,000 kilodaltons, which would be ultra-large and hyperactive VWF multimers. So in endothelial cells, the lower molecular weight VWF tends to be secreted both or constitutively, both basolaterally into the subendothelial matrix, which is the layer underlying endothelial cells, as well as apically into the blood vessel lumen, whereas larger molecular weight VWF tends to be further packaged into tight tubules, which drives the formation of Weibel-Palade bodies, which are right here. And these are rod-shaped organelles that can release their contents into the blood vessel lumen upon activation. And so endothelial cells release von Willebrand factor antigen and propeptide. Okay. And propeptide into the blood vessel lumen in equimolar ratios. And subsequently, higher molecular weight VWF can then be cleaved into smaller and less active VWF subunits by the metalloprotease ADAMTS13. And ultimately, von Willebrand factor has a half-life in the plasma of roughly 12 to 20 hours, after which it can get cleared by different receptors on sinusoidal endothelial cells, macrophages, and hepatocytes. So von Willebrand factor has two main functions in the plasma. So for one, it helps to initiate coagulation at sites of vascular injury by binding to collagen in the exposed subendothelial matrix, allowing for the recruitment, activation, and aggregation of platelets to help form the initial platelet plug. And also, as you can see, von Willebrand factor binds and stabilizes circulating factor VIII, which is an important protein in the coagulation cascade. And this prevents it from degradation and allows it to serve as a cofactor for the activation of factor X, an essential step in the development of a stable fibrin clot. So clearly, von Willebrand factor has an important role in the maintenance of functioning hemostasis. So when it's dysregulated in some way, it can have serious implications. So for example, on one end of things, on the left here, there's a condition known as von Willebrand disease, which is actually the most common bleeding disorder. And patients with von Willebrand disease can either have dysfunctional or very low levels of VWF, resulting in severe bleeding sometimes. And on the right, we have TTP, or thrombotic thrombocytopenic purpura. And this is characterized by low levels of the metalloprotease ADAMTS13. And so this then results in a high level of ultra-large VWF multimers in the circulation, which are hyperactive. And then this can result in an increased thrombotic risk, and in severe cases, organ damage and even stroke. So many different things are involved in the regulation of VWF at different phases of its life cycle, one of the more well-known actually being pregnancy. So pregnancy has long been associated with an increased risk or increased thrombotic risk, as pregnant women are 4 to 5-fold more likely to develop venous thromboembolism. And this is due to a few different reasons. So about 30% of this risk can be attributed to a reduced blood flow rate caused by the enlarged uterus, compressing the pelvic veins and forcing the blood to stay in the legs rather than flow upward. And this can then result in the accumulation of procoagulant proteins that can result in thrombosis. About 20% of this risk can be due to vascular injury caused by increased venous pressure and inflammation, which can damage the endothelium and again, increase risk of clotting. But the majority of this risk, about 50%, can actually be attributed to an induced procoagulant state or a hypercoagulant state in preparation for the blood loss that's experienced during birth. And this manifests as a decrease in anticoagulants, including protein S, and an increase in blood clotting factors, which includes but is not limited to fibrinogen, factor VIII, and von Willebrand factor. So these three factors here can actually all influence each other. And this hypercoagulable state is in part attributed by the mechanical changes that occur during pregnancy. However, there is a significant change in the hormone profile during pregnancy that's thought to have a significant effect. So throughout pregnancy, we see a big change in many hormones that function to develop the fetus and prepare for birth. Some of the main few being HCG, which increases in the first trimester and then drops thereafter. Prolactin, which progressively increases throughout pregnancy and remains high after birth. And then progesterone and estrogen. So progesterone and estrogen are interesting because they increase progressively throughout pregnancy and drop off just prior to birth. And when we look at von Willebrand factor levels throughout pregnancy, we see a very similar effect. So in this study from 2014, they found that circulating von Willebrand factor antigen here in red, factor VIII here in green, and to a lesser extent, von Willebrand factor propeptide here in blue, all progressively increased throughout pregnancy. But interestingly, they also found that the ratio between von Willebrand factor propeptide and von Willebrand factor antigen actually decreased in the plasma, which can be seen in purple. And so, as I mentioned before, the VWF propeptide and antigen are released into the plasma in equimolar ratios. So a decreasing propeptide to antigen ratio is actually indicative of an increased half-life of VWF antigen. So overall, these results suggest that pregnancy may be involved in either an increased von Willebrand factor production or even maybe a reduced clearance effect. And this also caused people to wonder whether there were other states of hormonal fluctuation that could also result in similar changes in von Willebrand factor. So when we look at hormone fluctuation throughout the menstrual cycle, we can see that both FSH and LH remain relatively constant besides a peak during ovulation, but that both estrogen and progesterone retain higher levels during the luteal phase when compared to the follicular phase. And some studies have actually shown that von Willebrand factor levels do fluctuate throughout the menstrual cycle, with on average a 10% increase in von Willebrand factor levels during the luteal phase compared to the follicular phase, when estrogen and progesterone are both lower. However, some studies found the opposite effect and others found no association at all. And in order to look at more isolated changes in estrogen and progesterone and how they might affect von Willebrand factor levels, we can look at hormone replacement therapy. So there are many different types of hormone replacement therapy, some of which require a continuous daily dosage of synthetic sex hormones, which is what I have shown here, and others which have more periodic dosages, but they all function to increase the average level of circulating synthetic sex hormones after endogenous hormone production decreases significantly during postmenopausal years. And some studies using combined HRT have found no association with von Willebrand factor levels. However, one study found that estrogen-only HRT increased circulating VWF levels after 4 weeks of usage compared to placebo group. So, all of this data indicates that estrogens may play some role in von Willebrand factor levels. However, there's little known regarding the extent of this relationship. So to put everything into relative context, in pregnancy, we see a progressive increase in the circulating estrogen levels throughout each trimester, starting at 200 to 2,500 picograms per mil in the first trimester and reaching levels upward of 7,000 picograms per mil in the last trimester. And likewise, we see an progressive increase in von Willebrand factor levels as well throughout each trimester. And when we look at the menstrual cycle, we can see that on average, circulating estrogen levels are higher during the luteal phase when compared to the follicular phase. And again, we see that studies have shown von Willebrand factor levels to be higher during the luteal phase when compared to the follicular phase. And lastly, when we look at hormone replacement therapy, we can see that postmenopausal women taking hormone replacement therapy have higher levels of circulating estrogen-like compounds when compared to postmenopausal women not on this therapy. And a study has shown that estrogen-only HRT has increased circulating VWF. But what exactly are estrogens? So I'm sure everyone in this room has a general idea of what estrogen is, but the term estrogen is actually an umbrella term for a group of steroid hormones that are derived from cholesterol and are capable of diffusing through cell membranes due to their hydrophobicity in order to exert their effects. Now, out of all of the endogenously produced estrogens, which I have up here, 17-beta estradiol is the most potent and can bind estrogen receptors with the greatest affinity. It's also the predominant estrogen during premenopausal years, throughout pregnancy, and the synthetic hormones in hormone replacement therapy tend to be modeled after its structure. So it's going to be the one that I'm focusing on for the rest of my talk today. However, all of these estrogens have a broad spectrum of physiological functions, some of which include reproductive regulation, bone density regulation, cholesterol mobilization, and inflammatory control, and also vascular functions, which I'll touch on today. And they have different estrogen receptors within different cell types that facilitate these effects. So there are 3 different estrogen receptors, which are placed into 2 different categories. So on the left, we have the nuclear estrogen receptors, and on the right, we have the G protein coupled estrogen receptor. And so the nuclear estrogen receptors are composed of ER alpha and ER beta. And these are both intracellular proteins that follow a more classical mechanism of estrogen signaling, in which they become activated either by binding their ligand or by phosphorylation, causing them to then dimerize and move into the nucleus to bind estrogen response elements and alter DNA regulation. And on the right, and these 2 proteins are relatively similar, with a 95% sequence homology in their DNA binding domain and a 55% sequence homology in their ligand binding domain. Now on the right is GPER, which follows a more non-canonical mechanism of estrogen signaling and was characterized much more recently than the nuclear receptors. And this is a transmembrane protein, which typically lies within the membranes of intracellular organelles, like the endoplasmic reticulum, but it's also been found in the plasma membrane as well. And once bound to estrogen, it can activate numerous different signal transduction mechanisms, a few of which I have listed here, which ultimately go on to activate rapid signaling within the cytosol or activate transcription factors to then bind DNA and regulate transcription. Now, it's also important to note that these 2 different estrogen receptor signaling mechanisms are not mutually exclusive. And so the nuclear receptors have been shown to associate with downstream GPER mediators to either aid in rapid signaling or to aid in transcription factor binding to DNA for gene regulation. And vice versa, downstream mediators of GPER can activate the nuclear estrogen receptors to then bind their response elements and alter DNA transcription. So with regards to endothelial cells, which are again the cells that form the inner lining of the blood vessel walls, it's generally accepted that they do contain GPER. However, there's a large amount of inconsistency within the literature regarding whether or not they contain either or both of the nuclear estrogen receptors. So one study from 2005 found that both estrogen receptor alpha and beta were undetectable in endothelial cells isolated from 7 different human umbilical cord veins. And the study from 2008 found that their HUVEC expressed estrogen receptor beta, but not alpha. And in 2014, this group found that their HUVEC expressed all 3 receptors. So despite these inconsistencies seen within this cell line, there have been several groups to demonstrate the effects of estrogen on endothelial cells, specifically with regards to von Willebrand factor. The first of which was conducted in 1984 and found that HUVEC exposed to increasing concentrations of estradiol for 5 days dose-dependently increased their VWF released into the cell media, suggesting that estrogens may play a role in the increased von Willebrand factor production. And while the study was conducted in 1984, there's been little to no additional research in this area until about 3 years ago, when this group assessed von Willebrand factor exocytosis in HUVEC after just 1 hour of exposure to increasing estradiol concentrations. And they found, again, that von Willebrand factor secretion dose-dependently increased with estrogen exposure in these cells. So all of this goes to show that estrogen does appear to regulate von Willebrand factor in some way, but we don't exactly know how. And it could be through some combination between direct estrogen receptor genomic signaling and indirect signaling cascade mechanisms. So taking all this into account, we hypothesized that estrogens increase circulating VWF levels through both direct and indirect genomic signaling mechanisms. And to address this gap in knowledge, our objectives were to investigate the effect of estrogen on von Willebrand factor production in vivo using a mouse model, also to confirm the previously found increased endothelial cell von Willebrand factor synthesis in response to estrogen exposure, and then lastly, to elucidate the mechanistic basis behind this effect of estrogen on endothelial cell VWF regulation. So we attempted to investigate the estrogen-induced von Willebrand factor production in vivo through two main studies. So for one, we wanted to verify whether VWF elevation seen in human pregnancy is recapitulated in C57BL6 mice, and if so, to then elucidate the mechanisms behind this increase by manipulating the estradiol levels in this mouse model. So in our mice pregnancy study, we followed 4 non-pregnant mice and 9 pregnant mice throughout the course of their pregnancy, which is roughly 3 weeks. And we collected weekly retroorbital plasma samples. As close to pregnancy term as we could get, we collected lung tissue from about half of these mice, and then we endpointed the remaining half 1 week later to see how von Willebrand factor levels change after pregnancy. And using our retroorbital plasma samples, we measured both estradiol and von Willebrand factor antigen. And using our lung tissue, we analyzed VWF mRNA by qRT-PCR. So on the left of this slide is the estradiol ELISA results from the retroorbital plasma samples. And as you can see, estradiol does seem to progressively increase throughout pregnancy, throughout mice pregnancy, becoming statistically significant by week 2, and then dropping off in the hours prior to birth, which is similar to what we see in human pregnancy. And then looking at the right side of the slide, we have our von Willebrand factor ELISA results from the same retroorbital samples. And we can see that at mice pregnancy term, which is right around 3 weeks, there's a surge in von Willebrand factor levels, which then drops off 1 week later. And the lung VWF mRNA data tells a similar story, with mice at pregnancy term transcribing a significantly higher amount of von Willebrand factor mRNA, an effect which then drops off 1 week later. So this all shows that the pregnancy-induced VWF enhancement is in part regulated by increased VWF transcription. So in our estradiol manipulation experiment, we had 5 different experimental groups. So we removed the ovaries from one group, which are the primary estrogen producers in the body, in order to reduce circulating estrogen levels. We then had a sham ovariectomy group. We had 7 normal mice, which we did nothing to. And then we administered both low dose and high dose 21-day slow releasing subcutaneous estradiol pellets into 2 different groups of mice in order to increase circulating estrogen levels. And we took weekly retroorbital plasma samples. And after 3 weeks, we endpointed our mice for lung tissue collection. And just like in our pregnancy study, using our retroorbital plasma samples, we measured both estradiol and von Willebrand factor antigen. And using our lung tissue, we looked at VWF mRNA by qRT-PCR. So here we can see that both low-dose and high-dose estradiol treated mice do have significantly higher levels of circulating estradiol, which become significant by week 2 and then drop off at week 3, presumably because the 21-day slow-releasing pellets are maybe running out by week 3. But interestingly, when we look at the ovariectomy group, which is in pink, we can see that although estradiol levels appear to look lower, this difference is not significant, suggesting additional involvement of non-ovarian E2 sources. So estradiol can be produced by a few different places in the body, one being adipose tissue. And when we look at weight gain over the course of the 3 weeks, we actually see that the ovariectomy mice gained weight at a quicker rate than the sham mice did, suggesting that these mice may have adapted to their low supply of estradiol by producing more fat, possibly. And when we look at our week 3 lung tissue mRNA data, we see a dose-dependent increase in von Willebrand factor transcription with increasing estradiol. However, just like our ELISA, or just like our estradiol ELISA, there's no significant difference between VWF mRNA production in our sham and ovariectomy groups. Very surprisingly, though, von Willebrand factor antigen data from the plasma or from the retroorbital plasma samples was the complete opposite of what we expect, which is that our high-dose estradiol-treated mice have the lowest level of circulating VWF, closely followed by our low-dose estradiol-treated group. Now, since these results are the complete opposite of what we expected, we actually repeated the study to make sure that thing was going well, and we got the exact same results. So indicating that this is likely what is happening. So these contradictory results show that estrogens do in fact increase transcription of von Willebrand factor. However, it's unclear what happens to the mRNA after it's transcribed, and it's also possible that von Willebrand factor may have something to do with the accelerated clearance of von Willebrand factor in the plasma. So we plan on doing a couple of follow-up analyses. For one, we plan to do VWF propeptide ELISAs so that we can look at the propeptide antigen ratio, which, as I mentioned before, a decreasing ratio is indicative of a decreased clearance effect of VWF. And also, we want to analyze mouse tissue RNA by qRT-PCR to look for different VWF clearance receptors to see if they've been upregulated in any way. So to test my second objective, which is to confirm the previous finding that endothelial cells do in fact increase von Willebrand factor production in response to estrogen, we first wanted to characterize the estrogen receptor profile in human umbilical vein endothelial cells, or HUVEC, through both an mRNA and protein analysis. And also we wanted to analyze the effect of estradiol on von Willebrand factor mRNA production in these same cells through both a dose response and a time course analysis. So, human umbilical vein endothelial cells, otherwise known as HUVEC, are primary cells isolated from the umbilical cord vein, as the name suggests. And we use these cells for a few different reasons. So for one, they're the most widely used cell type for in vitro endothelial cell research. They're also relatively generalizable to other endothelial cells, and they produce a large amount of VWF, making them a prime target for von Willebrand factor research. And just to remind everyone about the estrogen receptors that we're dealing with here. So on the left, we have ER alpha and ER beta, and these are nuclear receptors which can bind DNA directly to increase gene transcription or decrease gene transcription. And then on the right is GPER, which can either activate signal transduction mechanisms or transcription factors to then bind DNA and regulate transcription. So we first looked at the estrogen receptor mRNA expression profile in our HUVEC compared to our positive control, which were MCF7 breast cancer cells. And I'm sure many of the people in this room know, but just to go over, cycle threshold number represents the number of qPCR amplification cycles required to detect a target RNA. And so lower values tend to indicate a greater presence of target RNA, with values greater than 33 or 34 tending to be considered not really present. So when we look at our HUVEC, we can see that they contain little to no nuclear estrogen receptor expression at 34 and 35 CT values, but they do appear to contain some GPER mRNA. And when we look at the estrogen receptor protein through our Western blot analysis, we see kind of a similar result, which is that our HUVEC seem to produce little to no nuclear estrogen receptor protein. However, there is a faint band corresponding to where ER alpha should be. And we can actually see somewhat two bands that correspond to GPER. So that very low faint band at 42 kilodaltons is said to be the monomeric form of GPER. And then the higher band at 100 kilodaltons is said to be the glycosylated form of the receptor. Now, going back to this paper from 1984, which found an increase in von Willebrand factor production in response to increasing estradiol concentrations. This paper was published so long ago, so we first wanted to repeat it and try to reproduce these results before trying to elucidate the mechanisms behind why this could be happening. So we attempted to repeat the study in HUVEC by seeding down our HUVEC, replacing the media with estradiol, using concentrations ranging from 0.1 nanomolars, which corresponds to what you might see in a postmenopausal woman, or all the way up to 100 nanomolars, which you would see in very late-term pregnancy. And so we then incubated these cells for 5 days, changing the media every 2 days, after which I collected both RNA and protein for von Willebrand factor mRNA quantification by qRT-PCR, and for VWF antigen quantification by ELISA. And we also wanted to quantify estrogen receptor beta mRNA because there were reports that estrogen receptor beta actually increases transcription in response to estradiol. So we wanted to use that as a positive control. And after 5 days, we can see no significant change in both von Willebrand factor and estrogen receptor beta, which was meant to be the positive control. And when we look at protein, very similar thing. We see no real change in von Willebrand factor antigen in both the media and the lysate of our cells. But since our positive control didn't work, we thought that maybe 5 days was too long and we could have been missing the effect. So we decided to repeat the study, but for 3 days instead of 5. But still, we see no significant change in both VWF and ER beta mRNA. And again, looking at the protein, we also see no significant change in von Willebrand factor antigen in the media and lysate. So then this made us wonder if the estradiol itself wasn't working, perhaps due to any degradation in the media throughout the incubation periods. So we took a step back and tried to optimize for the timing of these experiments. So I seeded down HUVEC onto 4 6-ball plates. I then replaced the media with the same concentration of estradiol, which was 10,000 picograms per mil, which again corresponds with late-term pregnancy. And I incubated each plate for either 12 hours, 18 hours, 24, or 48 hours, after which, again, I collected the RNA and the protein and analyzed for VWF mRNA and protein. However, this time we also switched our positive control, just in case something about our positive control wasn't working last time. So this time we decided to use endothelial nitric oxide synthase, which has been shown by numerous different groups to increase in endothelial cells in response to estrogen. And interestingly, preliminary data from just one study shows a surge in both von Willebrand factor and eNOS mRNA production after 12 hours. However, this effect begins to decrease after 18 hours. After 18 hours and is completely gone by 24 hours. And when we look at protein from the media and the lysate of our cells, we don't really see any significant changes, but there does appear to be a slight increase at 18 hours. So this data suggests that there may be a decrease in estradiol's effectiveness after 12 to 18 hours of incubation. So in follow-up studies, I'll aim to replace the media more often in order to hopefully eliminate the impact of potentially the estradiol not working in the media, and to eliminate any dysfunction with the actual experiment, and also to hopefully prolong the increased mRNA transcript long enough to see it reflected in protein synthesis. So to assess our last objective, which was to elucidate the mechanistic basis behind estrogen's effect on endothelial cell VWF regulation, we first aimed to do an in silico analysis of the von Willebrand factor promoter region to identify estrogen response elements. And also we aimed to inhibit these estrogen receptors to determine which pathways regulate the estrogen-induced VWF synthesis in HUVEC. So between these two, we've only gotten to the first point here. So this image was generated using the JASPAR predicted transcription factor binding site on the UCSC genome browser, which is a website that provides genomic sequencing data. And JASPAR binding scores greater than 400 indicate that there's a likelihood of actual binding at that site with a p-value of less than 10 to the negative 4. And as you can see, a few kilobases upstream of the VWF transcription start site, we do find a couple estrogen response elements for both ER alpha and ER beta with binding site scores of over 400. And although these receptors are these receptor binding sites are relatively far upstream, since many promoter regions tend to be about 1 kb upstream, it's possible that the estrogen receptors act as either enhancer or repressor proteins for the VWF gene. So in terms of future directions, for our first objective, we hopefully aim to analyze the von Willebrand factor propeptide antigen ratio in our mouse model to assess the impact of estrogens on VWF clearance. Also, we aim to eventually analyze the mouse tissue RNA for different VWF clearance receptors to see if the expression of any of those have changed. For our second objective, we aim to optimize the timing and dosage for future estradiol cell experiments. And in our last objective, we aim to hopefully inhibit these estrogen receptors and observe the subsequent effect on von Willebrand factor production. And lastly, to mutate the estrogen response elements of any implicated receptor pathways in the VWF promoter region, and then to analyze the effects of E2 on luciferase reporter expression. And then lastly, I'd like to thank my supervisor, Dr. Lillicrap, for all of his support and guidance, as well as my supervisory committee members, Dr. Greer, Dr. James, and Dr. Nickel, as well as everyone in the Lillicrap lab, and Megan O'Donnell, who is in Ireland, but she's on the Zoom today. Oh, hello. And thank you all for watching.
Okay, so we got to remember to get people to repeat questions, or maybe I'll run around with the microphone. And I'll remember to turn around. So questions, go ahead, Paul. Here, give you the microphone.
Okay, that's a bad idea. That was awesome, Emily, and I think Dr. Nickel will back me up on this. Since your committee meeting, you've done a ton of work. And you actually took a whole bunch of our suggestions and did them. And to my enormous pleasure, the story got clearer.
Yeah.
I feel. So thank you and congratulations. I have a dumb question about HUVEC. So as you were presenting about what receptors HUVEC express or don't express and how they respond to estrogen in culture from the point of view of von Willebrand factor, I was trying to think about what do I think they should, human HUVEC should do. Like thinking about what the umbilical cord does and the cells that line the umbilical cord growing a fetus during pregnancy where estrogen is steadily going up. From an adaptive point of view, I was trying to think through, is there something that I would think would make more sense about the way those cells would respond or not respond? Is that something you've given any thought to? Do you have any comments you could make about that?
So in terms of, I guess, so would you be asking how you would expect HUVEC specifically to respond to estrogen?
Yeah, because it's a specific kind of endothelial cell that grows a human being under the influence of rising estrogen over time. So I just was thinking through, would I expect them to do something specific or not? That's like a 30,000 feet question. But.
Well, I know that estrogen does have a ton of alternative vascular functions besides just VWF, so it plays a very important role in vasodilation. So even just by producing enos. So that could be part of it. I think specifically part of the reason we're using HUVEC is some of the reasons I listed is they are generalizable compared to other really specialized endothelial cells like sinusoidal endothelial cells that have alternative functions, whereas just using HUVEC tends to be more generalizable to different cell lines. But in terms of what estrogen would do specifically in the umbilical cord vein, I think probably a large part of that is vasodilation. So that would be my best guess.
No, and that makes sense. And I guess what I was thinking about was if HUVEC responded in a very positive way to estrogen, that might not be a good thing in the long run in terms of fast blood supply to a fetus that's growing. So that's a good answer. Thanks.
Who's that? No.
Very, thank you. So in your mouse models, you saw estrogen treatment for estradiol increases VWF production, but not plasma concentration. Is there something besides constitutive release that stimulates that release into the plasma that you're missing?
So yeah, so there's two types of ways VWF can get, or two main types, I guess. They could be constitutively released, but usually the bigger VWF tends to be packaged into these Weeble-Palade bodies, and stimulation of that organelle does release von Willebrand factor into the plasma. So that, and other people have, I guess, looked at that as well. So I do think that is part of the picture. Let me just find it. So yeah, ideally, that's likely what's going on in this figure because they only exposed estradiol or HUVEC to estradiol for just 1 hour. So in reality, that's not really enough time for DNA transcription to happen and then for that to be translated into protein. And So this is likely due to activation of the Weeble-Pallady bodies. And yeah, there are several things that do activate Weeble-Pallady bodies, like aldosterone, epinephrine, things like that. And I think, yeah, these people suggested that estradiol is one of them. So I think this is also part of the picture.
Can I just follow up on that? Because
Mhm.
I was gonna ask about that too. Have you tried looking at Weeble-Pallady bodies in any of the endothelial cell systems you've looked at and what they do in response to estrogen?
Yeah, well, we did do a couple of shorter term experiments like this. I didn't really know where it fit into the presentation, so I haven't included it here. And we kind of left that be a little while ago, but yeah, we did do some short-term 1-hour estradiol experiments, and there did seem to be an increase in in the VWF in the media after that hour.
I'm wondering if you did it by microscopy and actually
Oh.
looked at what happens to the Weeble-Pallady bodies. Can you see evidence that they're actually disgorging content? Because the magnitude of this effect doesn't seem to be huge. I mean, it's not even twofold. So would you expect if you stimulated Weeble-Pallady body release from a culture of endothelial cells that you'd get more, a higher increase in what you found in the media at that point?
Wait, sorry if you.
Yeah, so this is the level that they're seeing in cultured HUVECs.
Yeah.
But, if those cells are actually full of Weeble-Pallady bodies and they actually disgorged a lot of that content, would you expect to see more than that level of increase, like instead of a twofold or less than twofold, maybe a tenfold level or something like that?
Well, I guess it depends on the strength of whatever agonist you're using. So I know that they use positive controls, histamine and thrombin, which are physiological, and they do increase the release to kind of, you can see a similar amount as the 1 micromolar, although that's not really a physiological concentration of estradiol. But yeah, if you were to use, I think it's PMA, you would see a huge increase. And when I was playing around with this stuff, I did use PMA a couple of times, and it was much higher, but PMA I don't think would be physiological. That's just something that you can use.
So the reason I'm pestering you about this is because I know that Dr. James over there has beautiful IF images of endothelial cells. And it seems like it would be, if anything, if nothing else, eye candy to see an effect on this, let alone the precise quantitation of it. But the other part of it that I find intriguing is the possibility that some of the effects that you're seeing aren't in the transcription translation aspect. It may be more in the trafficking part of it. So that wouldn't be surprising if that. So how are you going to address that issue of trafficking other than just looking at what's in the media?
Yeah. Well, I think that in terms of microscopies, that would be one way. But also there are 2 main ways that Weeble-Pallady bodies can even release their contents. So one is an increased intracellular calcium, and other is cyclic AMP. So even, I guess, somehow measuring levels of that intracellularly could maybe partially indicate what pathway might be increasing the Weeble-Pallady body release. Because yeah, there are many different signaling transduction cascades that are involved. But yeah, and also even using microscopy, you can, because based on whether it's calcium or cyclic AMP that's causing the release, it actually results in a different type of release of the Weeble-Pallady bodies. So one has more to do with like a physical squeezing of the Weeble-Pallady bodies.
Describing it in your proposal.
Yeah. And the other one has more to do with
it out versus
more like ionic, gradients and things like that. So yeah.
Other questions?
But just to pick up on that, I think Dr. Greer is making a really good suggestion that one of the other things you could do with conformal fluorescence is co-localize an endothelial cell or an ER marker, for example. So if there is a trafficking defect, you might be able to sort out where it's happened at. And I think that's an important thing to think through. If I had to bet, I might bet.
Might have to turn it on, Paula.
I think clearance is an important piece to look at as well, and it would be super interesting if clearance is like totally changed.
Mhm.
But those experiments that Dr. Greer is suggesting seem interesting to me.
Mhm.
Mackenzie can help you with that. Or calling your people on data.
You're on.
Thank you. I want to ask about the estrogen biology in human versus mouse. For example, are the levels of estrogen in blood comparable in mouses to human, and also among estrogens, for example, estradiol? Is it the same proportion of estrogens in mouse blood as it would in human blood? Is it also the predominant form? And just is the model comparable in that regard?
Yeah. So it is in terms of the actual concentration of estrogen, it's much lower in mice. But I think we partially took that into account. Like when we were figuring out dosages we wanted to give the mice, we had looked at other studies done in mice instead of thinking, oh, what's a high dose in humans and giving that to the mice. So yeah, definitely much lower concentrations. Even in pregnant mice, they'd probably not pass 1,000 picograms per ml. And in terms of the actual structure of the estradiol, I think they're definitely very similar. I know even for a lot of ELISAs, when you're measuring them, there you can, like one ELISA will measure human, mice, and it'll all be the same. So yes, I think they are the actual estradiol in the mouse and the human are very similar. And in terms of the receptors as well, and you didn't ask about that, but the actual receptors are very similar as well. No, we haven't talked before this. And in terms of the estrogen receptors, there's about like 90% sequence homology, and there's most of that homology lies within all the important regions. So the areas where it would bind things, like the DNA and the ligand and things like that. So yeah, estrogen and the estrogen receptors are very similar in both models.
So I will confirm Dr. James' point, so it does flow a lot better, and congratulations on all the extra work. I will support the microscopy piece as well, because I was thinking of the same thing, especially if you're also trying to think of which estrogen receptor pathway might be involved in that process. My question, though, relates to those 5-day experiments that you had. You mentioned a couple of times that you actually changed the media every 2 days. Did you, on the analysis part, it wasn't clear to me, did that include, presumably you collected the media that you were changing out to monitor, or did you only look at the 5th day media? And I was just wondering if you thought there might be any secretion in those periods of windows, and could you collect those and monitor?
Yeah, so we only did in the 5th day, but I guess that's just definitely something that we could do. So yeah, I added the estrogen, 2 days later replaced the media, 2 days later replaced, and then after 24 hours of incubation, collected that and measured it. But yeah, I definitely think that could be useful to see throughout the entire experiment how estrogen levels are changing.
Especially if something maybe secreting earlier, you're just missing it because you washed it out twice before you actually go to measure.
Am I allowed to ask? This is not a planting.
Oh, yeah, I heard the question.
Emily, don't worry, this is going to be easy.
Okay.
I promise you. Can you go to your hormone and pregnancy slide?
Yeah. Oh no.
Yeah.
This one.
No. Yeah, bring just the one prior to this. Yeah, this one here. Yeah. So in the mice, you see that in pregnancy, you recapitulate what you see in humans. The mRNA goes up and the protein goes up. But when you expose the mice to just estrogen, the message goes up, but the protein goes down. I mean, it really surprised us a lot. So looking at this, what do you think other things you might want to do?
I think going forward, doing both cell and animal experiments with perhaps maybe estrogen and progesterone in combination would provide valuable insight because it's possible. I mean, estrogen and progesterone specifically, there are a bunch of hormonal changes, but estrogen and progesterone tend to work together in a lot of other parts in the body, like in reproductive regulation. When estrogen is alone, it positively feedbacks to the hypothalamus to then release more FSH, LH, which then increases estrogen and progesterone. But then when you get a higher progesterone level and you have both of them that are high, they negatively feedback. So there are other circumstances in the body where when these two hormones work together, they cause an opposite effect of what they would cause when they were alone. So I think that in the future, I mean, besides doing the estrogen clearance experiments, also going back and maybe repeating the estradiol experiment, but using progesterone as well could show something different.
And are we sure that prolactin might have a role? I mean, presumably,
Yeah.
I mean, we know it's there for lactation purposes, but is there any chance of some sort of weird effect on?
There's a chance. I haven't ever seen that association. I also think the reason that my eyes are drawn to progesterone and estrogen is because they follow the same trend that BWF does. So it makes sense. But there's a lot of things in here that might actually impact von Willebrand factor, like even cortisol, that's a stress hormone. And it can bind to aldosterone receptors, which aldosterone increases VWF release. So I think there's a lot of parts in this picture that play a role, but it's just a matter of picking them out and seeing what each of them do.
Does anybody know what oxytocin would do to a VWF expression?
I don't think it would do anything to the VWF expression, not that I know of, especially just briefly there, but I thought it's a pregnancy hormone, so I included it in the picture.
Yeah. Sometimes they jam people in the picture.
I haven't looked at it systematically. I only looked at it in a patient or two. My impression of the data I've seen is that you're so maxed by the end of pregnancy in terms of your endothelium pouring out VWF. You can't jack it up any higher than DS.
I'm thinking the opposite actually, because, you know,
Oh, that
this,
it's coming down.
yeah,
Well, the peak of VWF though is a day or two after delivery.
yeah, just looking for something in there that might explain this bizarre observation that you're
Yeah.
seeing where it's going down instead of up. Is there some kind of feedback mechanism that you're overlooking in this that might be responsible for that?
The prolactin question is a big one actually, because clinically, the story we tell ourselves, and again, it hasn't been looked at very carefully, is that breastfeeding prolongs some of the coagulation changes that we see in pregnancy, and so that would have to be prolactin as opposed to ongoing progesterone and estrogen, which follows right away. So that's an important thing to think about.
Who's next? Oh, right there.
This is purely technical, so, for your objective 2, when you're measuring the VWF antigen, what type of media are you using?
For objective 2, so we used EGM media the whole time, but we used 2% FBS because when I started off using Optimum, but then I found that there was a lot of cell death. So and the problem with 20% FBS, which normally is what we do, is that FBS contains estrogen in it. So we reduced the estrogen in the media as much as possible, and then used that. Yeah.
I don't remember, did you use a positive control for those?
We used a positive control for the mRNA. Yeah. Well, we started off using ER beta. That didn't really seem to it either didn't work, or either the experiment didn't work or the positive control didn't work. So then we switched to endothelial nitric oxide synthase because we thought that was more of everyone unanimously agrees that in endothelial cells, estrogen increases eNOS. So it was a stronger positive control. So we did do that.
But not for the VWF secretion.
Not for the secretion. No.
That reminds me of a question about these sorts of positive controls. When you see these CT values up in the high 20s and into the 30s, are you really sure that signal is specific to the target? Is there something else that you've been able to do that would give you confidence that it's really the product?
Yeah, well, I think in terms of I've only really done QRT-PCR and ELISAs. From what I know, QRT-PCRs tend to be quite specific. So, and they kind of, the fact that not ELISA, a Western. So, do you mean for my for the estrogen receptors or for the VWA?
Any of these
market?
targets, but certainly the estrogen receptors, ER2, for example, and MCF7, is
Mhm.
that a real signal, or even GPER in
Yeah.
cells or HUVECs?
So yeah, even that was shocking to me to see how low they were expressed in MCF7 cells. I mean, they are still there, but I was expecting to see a little bit more. But the fact that it kind of does correspond with the
Yeah.
with the
That's
Western
correct.
kind of is like, because if you look at the MCF7 here, like
Yeah.
it's there, but
That's
very
a
light.
good point.
Yeah. So, and then you look at when you see the 21 for the MCF7, it's pretty low CT value, and then there's a pretty bright band. So yeah,
Yeah.
they kind of do seem to add up
Okay.
in my head.
This reminds me of another question about the positive control part. So especially for GPER, where you think you're getting downstream signaling from trimeric G protein, have you looked at other signal or thought of, I don't think you have, looked at other signaling proteins in that cascade, tried to assess their activation by estrogen receptor?
Yeah. So we have
Estrogen.
So we haven't done that yet, but that's definitely something that we plan on doing. I think we were kind of, again, in terms of elucidating the mechanisms behind why this is all happening, we were really just trying to confirm that it does happen first, which has proven a lot more difficult than we expected it to be 2 years later. But yeah, so that is definitely something that we're thinking of. I mean, I have ideas. I know that in endothelial cells, like which one is it? I think it's the MAP kinase
Yeah,
pathway.
and phospho-ERK is what
Mhm.
is the go-to for that, because
Yeah.
the reagents are so good.
Yeah.
If you can't get an activation with that, assuming it, I would assume that it should be activated by it, but
Mhm.
then you ought to be concerned.
Mhm.
Things have quieted down. It's 3 minutes to the witching hour. So
Oh,
Joy, pardon
I
me.
have a question if I could sneak in.
Sorry, go ahead.
Is that okay? Yeah. So going back to the media and controls and everything else, have you tried doing these in charcoal scrubbed serum to see if that knocks your negative control down enough to see an effect?
so we thought about it. We haven't tried it yet because we kind of thought that, I mean, just looking from other papers as well, that the cells don't seem to do too well in the charcoal stripped FBS, because charcoal stripping doesn't just remove the hormones, but it removes like fatty acids. It removes, I think, all nonpolar compounds, which the cells tend to like. So we didn't end up doing that. We just tried to reduce the media or reduce the FBS as much as possible in order to reduce estrogen as much as we could without like hurting the cells or causing them to act in a way that they normally wouldn't. But yeah, I mean, I'm considering all possibilities at this point.
So just to confirm for my own selfish interest, because I work on endothelial cells. So if you supplement your EGM2 with like the same amount of serum, but using the charcoal strip, they still don't do great. So even with all the other additives in there.
Well, so sorry, I don't know if maybe I was unclear. So we've been using 2% FBS, but not charcoal stripped FBS, just 2% regular FBS.
But yeah, so I guess what I'm trying to confirm is like when the cells didn't do well in the charcoal stripped, was that supplementing EGM2 with all the other growth factors?
Oh yes, that was in the paper that I found that I was reading about. Yeah,
Right.
we didn't do that. I
Great
read it.
talk, by the way.
Oh, thank you.
All right, I don't see any more questions in the Zoom, no more in the room, so join me in thanking for